Okay, here we go. Today we will discuss this paper, uh, Growth Promotion and Yield Enhancement of Peanut by application of plant growth promoting rhizobacteria. So I wanted to, rather than giving a lecture on plant growth promoting rhizobacteria, which is a pretty easy concept to understand, I felt like it'd be better use of our time if we analyzed a paper and you can actually see some of the data that shows how useful some of these bacteria are. Okay, so this is with respect to peanuts. I also felt like this would be a good lecture because Alabama is big for peanuts, right? So there's a lot of people in Alabama who care about producing peanuts. Okay, so we'll just go through some of the main points that I pulled out from the introduction, and if anybody has any questions, feel free to raise your hand and ask a question whenever you need to. Okay, so the interactions in the rhizosphere play a pivotal role in transformation, mobilization, solubilization from a limited nutrient pool in the soil. So the gist of the point here is that plants grow right in the soil, and the soil is actually a pretty nutrient scarce environment. There aren't really a ton of nutrients in the soil. So when you are near a plant, where do you think the most nutrients in the soil are? If you're like sort of like within the region near a plant. Yeah, right next to the roots. The roots actually produce what are called root exudates. Will somebody open that door? Root exudates. So roots actually secrete metabolites that bacteria can eat. And so interesting way to see the system is that the roots are actually secreting sort of food that the microbes that they like can come close to the roots. And so they're almost like the plants are actually almost like culturing kind of like their own little microbiome in the rhizosphere, okay? And the term rhizosphere, right? So the rhizosphere is this sort of region that is in close proximity to the roots, okay? And some bacteria will live in this region, and some bacteria are endophytes, and they'll actually like colonize and grow inside the plants, okay? And so the idea that this paragraph is suggesting is that there's actually some microbes that can mobilize and solubilize some of the rare nutrients. So things like nitrogen and phosphorus are often found in inorganic forms that cannot be utilized by organisms. And so there are some microbes that can take those resources, convert them into a more readily usable form, and sort of like feed those to the plant, okay? Let's see. Thus, the need of the hour is to enhance the efficiency of the meager amount of external inputs by employing the best combinations of beneficial microbes for sustainable agriculture production. So another big thing in agriculture, right, is the quality of the soil, nutrient content in the soil. And so there's kind of like a newer idea, well, sort of maybe not super new, but relatively new, that if you could just figure out like the perfect like microbial cocktail, you could put that near the seeds or as a seed treatment or in the soil, and you could plant that, and then your plants might do much, much better because they're in this microbiome associated environment where there are microbes producing everything they need, okay? So that's sort of like the idea of PGPRs. PGPR stands for plant growth promoting rhizobacteria, okay? Let's see, what else is said? While considerable attention has been given to the immense potential of using fluorescent pseudomonads as biocontrol agents against soil-borne fungal pathogens. Okay, so let's dissect this. So when they originally discovered PGPRs, the term PGPR was coined by Joseph Klepper, who was a professor in our department. Actually, he's emeritus professor right now, so he is retired. But he originally coined the term PGPR, and the original paper is something like 1980. It's either Nature or Science, can't remember. I used to teach that paper, but essentially the gist of the paper is Dr. Klepper goes out, I think it's on potatoes. He samples from some soil around potatoes, extracts some of the microbes from that soil, and he discovered a variation of Pseudomonas, which is a genus. So he discovered this Pseudomonas, which sometimes are just naturally fluorescent. And this Pseudomonas was a a bacteria that actually protected the potatoes from getting sick by plant pathogens. Okay. We'll talk a little bit more about that mechanism below. So understand that within PGPRs, there's sort of like different things that PGPRs can do. We'll enumerate that list. But one of the things that PGPRs can do is they can inhibit plant pathogens from taking control of the plant. Okay, next paragraph. Beneficial free living soil bacteria isolated from the rhizosphere, blah, 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 blah. PGPR, I introduced that. Okay, so different types of PGPR genera. These are things like Azotobacter, Azospirillum, Pseudomonas, Acetobacter, Burkholderia, Bacilli. Bacillus is a real common one. So we talked in the last lecture about Bacillus thuringiensis. There are BT PGPRs, and there's also things like Bacillus subtilis. Other Bacillus derivatives are commonly PGPRs. So there's essentially like a bunch of different genera that can do this. It's not just one particular bacteria. Let's see, the next thing. Okay, mechanisms of PGPR mediated enhancement of plant growth and yields of many crops are not fully yet understood. So there's sign of in many studies, people find kind of like these magical properties of PGPRs, and many of these mechanisms are not quite understood. So there are many studies here that they're citing. They're kind of listing things that they can do. They can produce hormones like IAA. This is essentially another way of saying auxin, which remember what auxin does? That's a plant hormone. Yeah, helps root formation. They produce sometimes cytokinins, so that's another hormone, shoot formation, nitrogen fixation. So this is kind of just listing things they do. We'll go more into this and giving you a bunch of citations if you wanted to read about these different effects. So there's kind of like these magical things that PGPRs can in theory do. Okay, but they say, but in peanut reports, reports on PGPRs are kind of scanty. What year is this? This is 2004. Okay, so this might be a little bit out of date. There's probably much more research on this since then. But at this time, they're saying more work needs to be done on finding PGPRs for peanuts. So it's probably worth talking about, in that sense, PGPRs, ones that actually work, are often specific to the type of plant. Okay, so for example, a PGPR that works really well on corn might not work really well on peanut, or a PGPR that works on cotton might not work on peanut, etc. So many times these studies proceed by starting with the idea that like we need to go out and we need to find PGPRs that are associated with the crop that we actually care about, and then we'll start from there. So many of these research studies start by going out into the soil around a field where they're planting a crop that they particularly care about, and then they collect that soil, and then they are finding bacteria that live in that soil that's associated with their particular plant. Okay, and then they talk, just sort of briefly mention kind of like a minor problem with planting peanuts is when you plant peanuts, they're saying that you get a high mortality of the seedlings. So here's a life cycle of the peanuts. When you plant the peanut, right, the sprout comes out after germination. And so they're essentially saying there's a significant mortality, seed mortality rate, that you lose some seeds that don't germinate, okay? And that problem might be solved by PGPRs. For example, if the mortality is caused by these plant pathogens like this Aspergillus niger and this Sclerotium rolfsii, which are fungi, you might be able to develop a PGPR that could protect against those soil-borne plant pathogens, okay? And so if you could do that, you might be able to get a higher germination rate, which in turn is going to lead to a higher yield. So that's kind of like one thing that they're thinking about. Okay, so they start the study. And again, the idea is they're going to go and they're first going to just sort of like collect, kind of like just what we did. Like we collected a whole bunch of microbes. They went out into the soil around their peanuts, probably in a previous study. They didn't actually, I don't think they actually listed the collection methodology in this study, but they have collected 233 potential PGPRs that they're going to test for various activities in the context of peanuts. Let's see. Just looking at this quick. Okay, so now let's go to the results. First table of the results. So here's where they take a bunch of these PGPRs. The first thing that they do is they screen them for, so let's say step one, they had 233. Here's what they say right here. Of the 233, they first subjected them to this screen for ACC deaminase activity. So I just quick pulled up what that was. The ACC deaminase leads to production of ethylene, a plant hormone involved in stress response. So essentially what they're trying to do is if they can get the production of this enzyme, like the overproduction of this enzyme, it essentially like bolsters the immune system of the plant, okay? And so if there are plant pathogens in the soil, it might be a great idea to sort of like bolster the immune system of the seeds right when you plant them so that they're kind of like sprouting out with this immune system bolus. Okay, so that's sort of the idea. And they're able to screen for this ACC deaminase activity. And here they actually, where is it at? Where's that data? I'm not seeing where they actually report that data, but they screened those 233 and they came out with like a list of like essentially like 8 to 9 that they're really excited about. Okay. And then those 8 to 9 PGPRs, that's these. Then they tested those 8 to 9 PGPRs for a whole bunch of different PGPR activities. And now it's actually worth going through and defining each of these. Let's see. So I defined the list here. So here's kind of like a list of PGPR capabilities. And we can go through and talk about each of these. Okay, so the first one is called siderophore production. This was in the original discovery of PGPRs, that Pseudomonas that Dr. Klepper found with potatoes. It protected the potatoes against plant pathogens. And the way that it did that was through the production of siderophores. Okay, so what actually that is, I have some more room over here. If imagine, here's the roots. Imagine you have a green PGPR, okay, and you have let's say a blue pathogen. Okay, so the idea of the siderophore production is the term siderophore is some chemical or some protein or some molecule that chelates iron. Okay, so if you have some, let's just call it S, some chemical or some protein, what does it mean to chelate? This is a chemistry term, chelate. Do you know what that means? I think you had it right. Say but louder. I don't know if I'd say surround, but bind and hold on to ions. Yeah. So to chelate means you're binding to and you're holding some particular ion. So these siderophores that were produced and secreted by these PGPRs, this Pseudomonas PGPR, it would bind to iron. Okay. And iron is kind of an essential nutrient that's rare in the soil. And so if the PGPRs are secreting a bunch of these siderophores, okay, in the soil, and each of those siderophores is sort of like chelating all the free iron in the soil, you essentially create kind of like a buffer zone where there's no iron. And it makes it difficult for pathogens to colonize that area and get close to the plant. So that was the original mechanism of the PGPRs, the siderophore producing PGPRs discovered by Dr. Klepper. This is called a bioprotectant. This is but there's various, these features fall under various mechanisms. I would call this a bioprotectant mechanism where it's protecting the plant against plant pathogens through the chelation of iron. Okay, that's siderophore production. IAA, that is what the long form of it. Did I put it? That's just auxin production. Okay, so that's just sort of like a different way to say that plant hormone. So hormone production for the plants. You can imagine if this is actually a very common thing that you see in PGPRs. I remember seeing the first picture that kind of blew my mind about PGPRs is, I don't have the picture actually here in this presentation, but I work with a colleague who studies turf grass. Okay, so imagine this is like turf grass, and if you put a side-by-side picture of minus PGPRs and plus PGPR turf grass, you look at the roots, so you can pull the grass up and you can see the roots. The roots of plus PGPR turf grass look like this. So you see a massive increase in the root length and the biomass production. And this can be very helpful for plants depending on the type of plant. And you can imagine how this would all be regulated potentially by microbes that were living in the soil. And if the microbes were able to produce extra auxin hormones, it's going to be essentially like pumping those hormones into the plant, and the plant's going to be producing a lot more root length. Okay, so that's one common mechanism of PGPRs specifically. That one is called the biostimulant. So this is when they're producing the plant hormones. This is the second one. That's a biostimulant mechanism. Let's see, go back to this list. Okay, so we covered that. Ammonification. So that's essentially taking nitrogen and making it more accessible. So here I have nitrogen compounds like amino acids and nucleic acids from dead cells and waste materials are converted into ammonia, NH3, or ammonium, NH4+. This conversion is a crucial step in the nitrogen cycle, making nitrogen available in a form that plants can absorb and use for growth. So just to understand that there are cycles of some of these inorganic elements like nitrogen, phosphorus, and calcium. And the nitrogen and the phosphorus and the calcium need to be in a form that can be acquired and readily utilized by the organism. And some of these microbes can do that. Okay, so ammonification is one of those. A second one is solubilization, so making this tricalcium phosphate more soluble. That's putting phosphate in a form that's solubilized that the organism can use. These would be the third mechanism, probably. The third mechanism is what's called biofertilizers. So we've talked about biostimulants, bioprotectants, and biofertilizers. Biofertilizers would be taking the nutrients and making it more readily absorbable for the plant. Okay, so those are three separate types of mechanisms. And then here are just 5, 6, 7, protection or inhibition against the growth of various plant pathogens, Aspergillus niger, Sclerotium ralphsii, and Aspergillus flavus. Has anybody cultured microbes? Has anybody ever seen Aspergillus? If you ever start to grow your own microbes or grow your own fungi, which is a fun hobby, this is one of the most common contaminants you'll see. It's everywhere. Aspergillus. It's like a black mold. There's also a green mold. I can't quite remember what the genus species of the green one is, but there's a ton of these that are also plant pathogens, and they're everywhere. And so again, if you could develop a seed treatment that would protect against some of these plant pathogens, which cause various diseases. So one of these causes southern blight disease. Fungal diseases are a pretty important area of plant pathology. Okay, so if we could develop PGPRs that protect against these, that would be very useful. So these are all the things that they test for in this study, in the original 9 PGPRs. And that's what this table encompasses. Okay, so this is actually a ton of work that they did that's condensed into a table. Okay, so the first thing that's probably worth pointing out is this, because they actually talk about this in one of the figures. And it's one of the things that I was talking about, root length of the seedling. So they're actually measuring the root length of the seedlings, and they're comparing these with a control. So the control receives no PGPR treatment, and this 6.03, that's the average root length of these seedlings. And you'll note that when they treat with these PGPRs, they get, all these are statistically significant. That's where you see the stars. So there's a statistically significant increase in the root length of the plants. Here, 8, 9, 7, 8, blah, 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 greater than 6. That's the gist. And they point out the ones that they're most excited by, which are sort of like the highest, PGPR 1 and 2, which are 8 and 9, 4, which is an 8, 7, which is a 9. So they kind of picked like the highest ones that are making the roots longer, and these are kind of like their favorite ones. And then they test for all these other things. So they test for siderophore production of auxin-like substances. Here's another thing they did. They tested for intrinsic antibiotic resistance patterns. So these are, here they're listing whether or not they're resistant against these antibiotics. If you see it, that's resistance that they detected. Why would they do this? Why would they test for antibiotic resistances? Thinking about synthetic biology and biotechnology in general, like why is this worth doing? Yeah, if you ever wanted to like engineer any of these or select for some of these in some, either maybe you wanted to give them a plasmid or maybe you wanted to somehow make it easier to culture them specifically so you could use an antibiotic, you would immediately need to know sort of like a profile of whether they have resistances to common antibiotics. So some of these have pretty substantial resistances. The 100, the superscript 100, that's probably the mgs per mil. Antibiotics are often given in concentrations of mgs per mil. So typically 100 mg per mil is a standard concentration for things like ampicillin and chloramphenicol. So that's what they're kind of like listing here. So many of these are already resistant against some of the main antibiotics, which would probably make it difficult to immediately transform them with some of the most basic vectors. So you'd probably have to build a plasmid that has a kind of like a unique selection cassette based on what they're seeing here and observing. And then here they have we'll actually look at the data of this below. Here they're testing whether they inhibit the aspergillus or the sclerotium. Okay. Anything else worth pointing out? Yeah, that's pretty much all of those worth pointing out with that table. Okay, let's see. Okay, now let's actually start looking at the data. So I thought this paper was actually pretty interesting to read. Some of these figures are really interesting. I thought these were really interesting to look at. So here is A. Let's look at this. What's being shown here? I kind of have already started to annotate. What's being shown in figure 1A? What are these things? Nematodes? Yeah. No, they're okay, so these are seedlings. So there's a seed that's like germinated. Okay, so we all see that. What's being compared? Yes. Okay, and what's the left and what's the right? Yep. So this is plus PGPR, PG. I think I can do this. PR, and this is minus PGPR. Okay. What's the visible phenotype? Yeah, so these are probably this is probably longer. What else do you see? Yeah, these like nodule things are more robust in the PGPRs. They talked about that a little bit in the introduction. They talk about how these, I think they're termed nodules. Again, I'm not a plant expert, okay? But these nodules seem to kind of like grow out into regions where the plant growth promoting rhizobacteria are making or solubilizing key nutrients, okay? So you get a better production of these kind of like nodule growth, which is going to in turn translate to higher nutrient uptake in the plants, okay? So I like this figure because it's really just like, again, I just like obvious things that you can just see, plus minus treatments, okay? So that's what A is. Okay, what's happening in B? What kind of experiment is this? It's that type of experiment, although I cannot tell. Okay, so some of this is hard for me to read, but you're exactly right that the type of assay is they construct it like a plate. Okay, so this is a bacterial or a fungal or a microbial like inhibition assay. And usually what you do is you create a plate and then you put maybe like a drop of something maybe like A, and maybe then around that you plate something like B, or there might be different ways to do this. Like maybe you will just uniformly like spread a spread and then like a what? A pill, a pill. So you would do a uniform spread and then you'd put like a drop or something. You'd put like an area of whatever you're treating or testing in the middle. And then you just check to see the phenotype. Yeah, this was hard for me to interpret because I could not tell. This is Aspergillus niger, which is black. So I think this is actually the Aspergillus. And I could not actually see what you would call inhibition. But they claim that there's inhibition in here. Does anybody have any other interpretation of this?
say blue, and then the center, the black part, would be the place where P-fluorescence kills.
In theory, that's what you would expect. Yeah, but I think that's incorrect because I think that the aspergillus is black, and I think that the P fluorescens, which is Pseudomonas, I think that's white. So I think they did so this is my interpretation. Sometimes data is really hard to interpret. My interpretation is they did a flat spread of P fluorescens and they put the aspergillus, but I don't see like a strong inhibition. You see a stronger phenotype here. Here's a better assay where they're doing something very similar, except it's set up in a different way. Here they did a streak of, here is the Pseudomonas, this thing. So it's like a toothpick smear of Pseudomonas. And then this is the fungus. This one you can actually tell, like that's obviously a fungus. How do you know that this is a fungus? Who knows basic biology? You can see the mycelium. So fungi have mycelium, which are these like long kind of like spiderweb like cellular structures that interconnect. So you can actually see the mycelium. That's how you know it's a fungus. Okay. And what happens to the mycelium as it's growing closer and closer to the Pseudomonas? Yeah, it stops. So here is like a much better piece of data that you can see where there is clear inhibition here, because right here is just a flat line where it's not growing past this. And what this implies is that the Pseudomonas is secreting something that's diffusing into the media that's preventing the fungi from growing closer. So I would say the data here support strong inhibition against this disease, which is the southern blight. But I don't really see strong inhibition against Niger, in my opinion. Okay, let's look at some of these other things. C. Does anybody remember what this was? Again, this is kind of like an older paper, so you're kind of like looking at like you probably would not see old black and white data like this anymore. But yeah, this is fluorescence. Why are they doing this? It's an interesting question. Why look for fluorescence? Does it matter? Like, is fluorescence going to contribute to a better PGPR? I don't think it's a control. I think this is just sort of like the culture of the field, where when PGPRs were first starting to expand, people really got excited about the Pseudomonas, and some of the Pseudomonas strains are naturally fluorescent. And so maybe, again, I don't know, but maybe in the field, maybe they thought there might have been some kind of distinction between like whether they're fluorescent or not, and maybe the fluorescent ones are better PGPRs or not. Perhaps that in the field was kind of like an area of debate. So they just thought it was worthwhile to test for fluorescence. But today, this is probably kind of like irrelevant. And this is not engineered fluorescence. This is just like natural fluorescence. So this piece of data doesn't really contribute anything strong, in my opinion. Let's see here. E. E, they say, is production of siderophore by Pseudomonas, PGPR. And they say halos indicate the production of siderophore in cast agar. So again, like I'm not an expert on these techniques, but what I like about these techniques is it seems like it's a pretty simple experiment where you just make a plate. And if you make a special plate with this special agar, if there's a secretion of siderophores, you apparently see this sort of like halo effect. And I definitely see there is like a halo effect. Okay. So I'll take their word for it. I'll agree that it's producing some siderophore. Excuse me. Okay. Here's one I actually agree. I can totally like see the phenotype again as well here. So here is F. They're checking for solubilization of tricalcium phosphate in the PGPR isolates. So here's a special plate where there's probably a saturated amount of some calcium derivative. Calcium is typically often white, okay? And so what you can see is that when they spot out some of the PGPRs on this plate, this one you can see kind of has this like clearer region of the media. Here, there's a clearer region of the media right around as well here, as well here. This one probably not so much. So this one is probably not producing or solubilizing the calcium phosphate. Oh, so you might be seeing the phosphate. I can't quite tell what is the white color, the calcium or the phosphate, but if you see this sort of clearing effect, that is the solubilization of the pertinent nutrient. Agree or disagree? Okay. So that's kind of like what I took from this. Again, it's kind of like I like this because it's sort of like easy data that anybody, if they wanted to replicate this, you could replicate it. So I kind of take them at their word. I trust them. And this to me is the most convincing. Like if you're changing the morphology of the roots, that seems pretty interesting to me. Okay. Next figure. Let's see. This is growth patterns of different PGPR isolates utilizing seed leachate of peanut cultivar. Well, okay, so what is seed leachate? Take a guess. Why do this experiment? What is this experiment? And maybe, okay, so maybe we'll talk about the data and then I'll ask you more why. On the y-axis, what are they measuring? Which is measuring what? Yes, it's optical density. Yes, it's the user spectrophotometer at 600 nanometers. What's actually being shown then on the y-axis? Like, what is this a proxy for? Yes, which is a proxy for, yeah. So essentially, like on the y-axis is number of cells. Okay, so you're seeing a growth curve of number of cells, which they're measuring by OD. Yes, okay, and the y-axis or x-axis is time. Okay, so they're measuring number of cells. And they're doing this in a specific media, which they say includes seed leachate of peanut cultivar JL24, which is the peanut that they like. So what are they actually testing? Like, what is the purpose of this experiment? Yes. So remember what I said in the beginning that if you have some plant and there are these roots, the roots produce exudates, like root exudates, and the root exudates are essentially like the food of the PGPRs that associate with that plant. So it will not be useful at all to discover some PGPR that has all these great properties, but guess what? When you put it in the soil, it cannot live and survive around the roots of the plant. Okay. So what they're doing here, just like Ben said, is they're testing whether or not these various different PGPRs can actually grow when the only food that you give them is just sort of like extract from these seed leachates. So it's testing the idea that they could, in theory, grow in the soil right next to the roots. And that's, in theory, like all they would need to grow. Okay. So as long as you're seeing an increase in the curves, which I think every single one is showing you an increase, then it's showing you that they're replicating. Okay. And if they are, okay, so they're replicating. So I buy this. Okay. So they can replicate, at least these ones can replicate with the seed leachate. Okay, so once they've done that, these people were pretty ambitious. So they went and they did pot trials and then they did field trials. So they first do these pot trials, which is, right, you have some kind of pot and you plant in the pot. Why do this? Why not just do a field trial? Yes, definitely more controlled. Also, it's just, it's probably less, probably cost less, probably less infrastructure. So it's kind of like before you go do a massive field trial, usually nowadays, maybe you do these pot trials in a greenhouse. So maybe you call this a greenhouse trial. Like it's pretty common nowadays to do a greenhouse trial, which is sort of like a small scale mini field trial before you do a field trial. Another reason to do the pot trials is because nowadays, maybe this was not the case in India. I think I think these are Indian researchers. Looks like it. Yeah, India. I should actually also just say their names. Day et al. From Gujarat, India. Okay, so I don't know 20 years ago in India what sort of like the regulations were on this kind of thing, but nowadays you can't really just take like bacteria that you're growing and do a field trial. You probably need to run through some kind of like EPA registration or something like that to start doing field trials. So it's always helpful to do like a little trial in a greenhouse. So that's probably what they're doing. Although I don't know if these are actually in the greenhouse or not, and they say rainy season, so probably outside. Okay, so during 3 years, so again, this is actually a lot of data, a lot of work that they did. So 3 seasons, they did these pot trials where they're mixing in different rhizobacteria. Looking quickly here at the results. Here's the table. Effect of plant growth promoting rhizobacteria on the nodulation, growth, and yield of the peanut cultivar in the pots. So what I wanted to look at when I looked at this this data and this table, and if you actually read the results are kind of boring to read and they're kind of lengthy, where they just kind of list like, here's what we found with this, here's what we found with this, here's what we found with this, and it actually gets kind of really long and hard to read. But when I look at this table, there are some things that I really would care about as a researcher. Like if I'm going to be working with a PGPR, what's the thing that you would probably most care about? Yes, like I don't care about any PGPR unless it's going to give me more peanuts than not. That's kind of like the only thing that I care about. So when I looked at this table, the things that I pulled out were the biomass per plant. So like, are you actually getting more plant tissue? And then the pod yield per plant. Okay, and so all the top controls, the top row is all the controls. And some of these are showing statistical effects. So this one is showing statistical effects, increase in pod yield. This one is showing increase in pod yield. This one. But let's make some notations about some of the patterns we see. So would you say if the control is producing 3.5 pods per plant, and with the PGPR you're getting like 4.5, that's kind of like a 25% increase. I think that's pretty significant. Like a 25% increase in the number of peanuts you get just because you dip the seeds in a bacteria. That's pretty good, I think. So that's pretty impressive. What are some other patterns that you observe looking at this data? Spend a few seconds just kind of like looking, like what do you note? What do you see? Oh, you're seeing biomass. So you're seeing if one's consistent. So PGPR1, for example, that gave increase in pod yield and also did biomass. Yes, I agree with that. So if this is increasing, you typically see maybe a pattern here increasing. What else do you see? In this one. Yeah, so this is one thing that you often see if you're doing like actual outside work. Outside work in agriculture you can have a lot of variation. So here you're seeing year-to-year variation where in 2000 they had something with the PGPRs like really hit and they got statistically significant increases for essentially every single PGPR. And the biomass increase here is pretty impressive. Some of the biomass increases here, like going from 13 to the highest, which is 18. I mean, it's not quite a doubling, but almost a doubling of the plant biomass. That's pretty impressive. But you are seeing, that was kind of one of the things that I wanted to say is you're seeing a lot of variation. You're seeing variation year to year, and you're seeing variation strain to strain. So for example, let's see if we can find one strain here's one strain that was significant in the later 2 years, but was not significant in the first year. So it's kind of like what's going on to make these things inconsistent? These are the pot trials right now. These are not the field trials yet. So these are in the pots. So you'd imagine it would be more consistent in the pot. At least you would hope for that. But that's actually one of the criticisms of PGPRs that's worth pointing out that you can actually see in this data is one of the criticisms of PGPRs is oftentimes the effects are pretty variable. And so in the future, I think one of the important things that this technology needs to work on is making effects very consistent. And one of the ways that people think that they could make them more consistent is if they could identify the mechanism. You could, for example, if you identify the mechanism that was specifically increasing the pod yield, you might be able to just then figure out if that's a chemical, then you just add that chemical. So there might be a phase of research where initially people are using PGPRs and then they source the mechanism to some protein or chemical, and then they actually switch utility to that actual chemical formulation. That might be what happens in the future to make them more consistent. Another inconsistency that you see, not necessarily in this data, but with PGPRs in general, is PGPRs are often said to be highly location specific. So if I isolated peanut PGPRs from Alabama, it's unlikely that those PGPRs would do well in India. Okay. So that's another thing with PGPRs is you often are working with like what might be like local isolates, and those local isolates might not do very well in other locations. Okay, so these are just some things we're talking about, looking at that data. I don't think that's what we're talking about. Okay, let's look at this. Figure 3, I have to refresh my brain here what we're seeing. Population densities of PGPR isolates in the rhizotic zones of peanut. Okay, so log number of cells. So this is exponential, right? Each number is going to be a different power. So understand that this is log number of cells. Okay. And let's see, this is patterns of the organism in the rainy season at 45 DAS. That's the key thing when you read the legend. Did anybody catch what DAS was? I forgot the S too. It's it's essentially like something like days after planting. I can't remember what the S stands for. Somebody could quick control find it. It might be. Did I write it down somewhere? Sowing. Yeah. Okay. So like planting. So it's essentially like days after planting. So why is this data important? So they're taking a read 45 days after planting or sowing, and they're showing you this data. What are some of the conclusions, and why do they do this? Yes. Okay. So that's I would say it as persistence. This is one of the sort of the golden dreams of the PGPRs. And every time I write a PGPR grant, this is what I pitch is the thing about insecticides or fertilizers is you kind of apply the treatment, but maybe that's a temporary effect. The hope and the dream of the PGPRs is if you consider the dirt sort of like this microbial environment, you could, in theory, colonize the dirt with the best PGPRs, and they might, maybe not forever, but they might in perpetuity continue growing in that environment. So you could establish like a super fertile soil on your own property that would just persist year after year after year. That is the dream of this PGPR idea. And so what they're showing you here is after 45 days, after the seed treatments, they are still detecting these different bacteria, these PGPRs, in the soil isolates, okay, in different regions. So here, black is rhizosphere, dark gray is rhizoplane, light gray is geocarposphere. People who studied soil physics probably understand more precisely what those terms are, but different regions of the rhizosphere. So they're detecting the bacteria and it's persistent. They do a similar thing here in figure 4. I'm trying to remember what this is. How is this different? Oh, here. Here they're measuring the same experiment, kind of pretty much, but they're testing over a longer period of time. So instead of 45 days, they're extending to 75 and 105 days and 90 days. And again, most of these, I mean, you see, so they're kind of like, you're seeing a little bit, maybe perhaps replication where they're replicating in the soil and then they're decreasing numbers, but it might be stabilizing. Okay. So you might be seeing persistent, stable colonization of the soil. And that's one reason why this data, this paper is particularly exciting, at least for me. Okay. And then I always like to see these things. We'll kind of wrap up here quick. Figure 5. This is where they're actually showing you, well, what are they showing us here? These are very interesting, like old school scientific pictures where they printed off their labels. I took a picture. What are they showing you here? Yes. So this is actually the yield. And here again, like this, whenever I see these things, I'm always just so impressed. This is minus PGPR. So it says it's hard to read, but it says uninoculated, and this is inoculated, minus plus. And I mean, this is essentially like a doubling of the yield, at least in this particular peanut. Here's a different one, PGPR4, minus, plus. You definitely see more peanuts. This is harder for me to understand. I think probably what you're seeing here is like more biomass, more robust plants. seems like this is plus PGPR. This is minus. It's real hard for me to read that. I think it says control. But here's just a quick picture that shows you some pretty impressive data, minus plus PGPRs. This is from the actual field trial. So they did then proceed to field trials. What did I write? I took a section from the methods because I think it is worth just looking at how they actually do the PGPR treatment. And here's the section that says how they do it. It says the seed was coated with charcoal-based PGPR cultures at some density. Okay, so this I think is important because this application methodology is extremely simple. Like there's seed producing companies that produce seeds, and seed treatments are a pretty common thing. So all they do is they produce a bunch of seeds, and all they would have to do is grow vats of these cultures, and then you just dip the seed in the culture, and then you disperse the seed to the farmers to plant the seed. So it's a very easy application. It's not necessarily like we have to grow millions of liters and then spray like millions of liters of bacteria onto your field. It's often just a seed treatment, which is pretty impressive. Okay. So I think that's a cool thing to point out. The ease of the application of this is often pretty easy.
All of the issues that the PGPR that you apply, it is not viable.
Yes. Yes.
Do you talk, like I know there's quite a few people that are in this business.
Yeah.
You know, of ways they found make them
Increase the viability. Okay, so let me just say for people who listen later, the question was, one of the problems is often viability of the PGPRs long term. Sometimes they might die out. Is there any research being done on increasing the viability? I know that yes, there are people who research that. In fact, I have a student who's interested in that. One of the things that would be highly important is if you could take a PGPR and you could improve its metabolism so that I could more efficiently utilize those like root exudates. That might be a way to make it persist. Another thing that's very important is niche competition. So in the soil, if you're trying to get some PGPR to sort of like outcompete other bacteria that are already in that soil, you might need to develop a system where you can sort of like remove what's there, or so you might envision a system where you could engineer a PGPR that could kill other microbes that it was trying to outcompete against. People might not like that, but these are some ideas. Like I think that's definitely an area of like hot research. If you could figure out a way to make PGPRs more persistent, you'd probably make $1 billion. So there's definitely a lot of people who are interested in that. This is okay. So to wrap up, table 4 is the field data. So probably as I look at this quick here, probably what's worth looking at is the number of pods per plant and the pod yield. Here's the control. They are getting statistically significant increases for some of these things, but not necessarily for all the PGPRs. So you see some effect, but variability and inconsistency. I think that's probably the state right now of the PGPR field. Okay, any final questions about that? Okay, so we'll end there. PGPR, that's the gist of PGPRs. And